All right, welcome back, everybody. So today I thought we could take a final look at the spatial gene set enrichment analysis. And so if we remember what we had done last time, is we had looked at running the gene set enrichment analysis on the field of view that we have, all the different fields of view. In this case today, what we're going to do is we're going to take those scores and we're going to see whether or not those cells that have high gene set enrichment are spatially clustered more than we expect if they were just randomly distributed in our sample. So if you find this helpful, please let me know down in the comments. If you have questions, leave comments and questions. Otherwise, feel free to subscribe and like if it's helpful. Entirely up to you. Let's take a look at the function that we're going to try to emulate today. The function that we're working on writing for our particular data, since we have the data that we want to model anyway, is STenrich from the spatialGE package written by Dr. Oscar Ospina. What this is doing is essentially 30,000 foot view here. This first part is subsetting the data down to a particular field of view. So remember slide 1, field of view 1, for example. We're going to subset down our metadata and our expression data or our gene set data to include only those cells from that particular field of view. Next, what we're going to do is we're going to iterate over all of our gene sets. We're going to extract out those cells that have high, and in this case, it's whatever you select as being high. Here it's one standard deviation greater than the mean gene set score for that field of view. Subsetting out the cells that pass that threshold that you set. Then we're going to measure the distance between those cells in our field of view. If the distance of those cells is very, very small, meaning all of those cells are very close together, when we compare that to a same number of cells randomly selected from our sample, the distance between those, the observed cells, are those with true puppy. You can kind of see her. She's talking. Anyway, you'll be able to tell that those values being smaller means that the cells with high enrichment scores are more spatially clustered than we expect, because the permutations are telling us a null distribution of what would be expected by random chance, right? So we get our observed, and then repetitions here, or reps, is going to be the number of permutations that you set. And here the number of permutations is set to 1,000. So randomly selecting the same number of cells that are high 1,000 times from our sample, measuring the distance, we get that null distribution. So I think that's the gist of STenrich. You can take a look at the function that I have for our clear cell renal cell carcinoma Cosmics SMI paper. It's basically the same thing, just written for the Cosmics SMI data as we have here for this project. I further optimized it, as we'll see today, for the data that we have, and it's incredibly fast. So if you feel like the number of permutations of 1,000 isn't enough, you can bump that up to 10,000, and it should still run in a reasonable amount of time. I think I had 3 minutes and 45 seconds or something like that for all 393 samples, 50 gene sets, and 1,000 permutations. So it's very fast. So let's dive into the script that we have today that we're working with. So we have the functions that we had previously worked with, tidyverse, which allows us to do our piping. That way we don't have to assign a data set to a function, edit that data set, so on and so forth. We have PBMC apply, which gives us a progress bar, which comes in great handy when doing these large sample things. So we have an idea of how far along our analysis is. And then a new function that will build this heat map here down in the bottom right hand corner with is complex heat map. The metadata that we have is the exact same as last time. And just like last time, we have to add that column in for a unique field of view so we can subset our data by that. So we'll just pull out a field of view and then start working with that data. Finally, we have to import the data that we had created last time with the gene set enrichment analysis that we had saved for all field of view gene set enrichment analysis.rds. Next, we have to select or set some environmental variables that we'll use throughout the permutation approach. We have cores, so I have used 8 cores, but I don't think the environment's big enough to really impact this. We can set this to 16 if we have that available. It should work just fine. The number of standard deviations, I'm keeping the exact same as the STenrich default. The minimum number of cells, I think I'm even keeping the exact same. Yep, minimum units. And then the repetitions is the number of permutations. And like I said, with the method that I have here on 45 through 77, if you feel like you need more permutations, you get a more granular p-value for the gene set, spatial gene set enrichment analysis, feel free to bump that up a little bit. If you have a cluster, you can go hog wild and get a very accurate p-value. Some of them are borderline, which more permutations might help, but I think 1,000 permutations is fair. Next, we have our gene sets that we had last time. Remember, we imported that as all fields of view. Every single element is a field of view, and then we have 2 things inside. We have the cell level gene set scores, and then we have the phenotype level. What we're going to do is we're going to extract out the cell level gene set scores and then bind them together. So we're going to take all of these, every single field of view's cell sets, and we're going to create an incredibly large data table where each row is a cell and each column is a gene set with a score for that particular cell. Next, what I did is I just extracted out the unique fields of view from our metadata. That way, when we go on to do the spatial gene set enrichment analysis, can just subset right to a particular field of view, which by happenstance, we're just passing in. So these are going to be the function variable that we're using. So by passing these in, we get a variable called FOV, which is one of set names, which is one of these. So slide 1, FOV 100. We'll cat that. I think I have this, yeah. So what I'm doing with this is I'm just catting it. That way, if you're only using a single core, you'll see in the terminal that it's working on this field of view, and you'll be able to keep track of it that way. But with the progress bar, this doesn't come out, and it's not needed since we have the progress bar. The next thing that I'm doing is I'm subsetting the metadata to be only with this field of view. So for example, FOV equals slide 1 FOV 100. Slide 1 or a slide meta is going to be only the metadata from that field of view. And I'm selecting the X locations, the Y locations, and the cell ID. And I'm selecting that cell ID because what we can do is merge that with our gene set scores and get slide data, which has the X and the Y location, the cell ID, and then all of our gene set scores for those slides. And then we can use this data passed into another lapply function, which is this guy, and calculate the enrichment score, spatial enrichment score for each gene set. So we're going to iterate over every single gene set. GS will be one at a time. From our slide data, we're going to select the X locations and the Y locations, as well as the column of that gene set. Then we're going to take the mean, because remember we need to be able to set a threshold for what is considered high enrichment. We have the slide data. We're going to find the mean of that particular gene set for this FOV. We're going to find the standard deviation for the gene set in this FOV. And then high is going to be those cells that are truly considered high gene set scores for this FOV. And in this case, we have 59 out of the 323 cells have high from mean plus 1 standard deviation that we had set above, gene set scores. And then RS is a length that will help us later on, which is just the length of those high cells. Here I'm checking whether or not the number of high cells is less than that threshold that we set. So if we have less than 20 high cells, the distance that we're measuring for observed can be unreliable, as well as the permutations. So we just return an NA, and we can deal with that later. The observed is going to be the pairwise distance for all of the cells that are truly considered high. So by doing the distance matrix, so distmat here is a cell by cell for this field of view. We can take a look at that distmat. We have 323 columns and 323 rows. What we can do is subset to those high cells. So high is cell 3, 6, 8, 9, 21, so on and so forth, which will give us another table that is RS by RS. So let's look at just this. And then dim. Oh, I need to remove that. There we go. There we go. So we have RS by RS. And since we only want the distance between each cell, we're not going to count cell 3 to cell 5 and then cell 5 to cell 3. We just want the distance once. We're going to divide it by 2, which is going to remove either the lower triangle or the upper triangle of that matrix. And that's our observed value. Next, we can do permutations, which is essentially the same formula here, but instead of giving it the high locations on that FOV, we're going to sample the rows of this particular field of view. So we're going to sample 1 through 393, the same number of times, 59 times, and that's going to be the cells that are for this particular permutation. And then we can measure the distance and divide that by 2, and that's now our permuted distance. We can take the permutation distance. If the permutation distance is less than the observed, we're going to count that as a success, and then we're going to divide that by 1,000. So if we have 2 permutations that are less than our observed, we have 2 out of 1,000, and that's going to be our p-value, 0.002. And so we're going to return that, which ends up being p-value here. So the output of p-vals is going to be a vector of length gene sets. And so I wonder if I have that somewhere. P-vals, yeah. So p-vals, here we go. So for this field of view, we have the p-values that are associated with every single gene set. And this is spatially enriched gene set. And it looks like here, most of them are not spatially enriched. So here I have IL6 JAK-STAT3 being enriched. Now that we have all the gene sets for this FOV calculated, we'll do that for every single FOV, which is the entire wrapper here. And so results is a list of length FOVs. Now, since we have a list, we need to convert that to a data table, and then later on we can convert that to a matrix, as you can see here in the bottom right-hand corner, and plot that. So what I'm doing is I'm going through extracting out, so the names of our results, which will be our fields of view, extracting that out and then transposing it. So now we'll have our gene sets in columns, converting it to a data frame, and then adding another column at the very end, which will tell us which sample or which field of view, unique FOV, that this row came from, or these p-values and gene sets, p-values associated with gene sets had come from. And then by binding that all together, we get a really nice data frame where each row is a field of view, 392, and each column, except the very last one, is our gene sets. That very last column, like I had said, is going to be our unique field of view. All of the values in this row are associated with this field of view, and these are p-values for that gene set. And then I just saved it out. That way, next time we don't have to run through the entire iteration of FOVs, gene sets, permutations, that kind of thing. We can work with this in the future. Finally, I thought making a plot is the easiest way to visualize our data. And by doing this, we can see some trends which are kind of standing out already. in the data that we have for spatially enriched gene set analysis. In order to use the complex heat map package in the function heat map, we have to make sure that what we're passing in is just a matrix of numbers. So that last column that we have on there that's telling us which field of view that the gene set p-values are coming from, we actually have to put that as the row name or remove it entirely if we don't care about the sample and we just want to look at global trends. So what I did is I created the row names from the column, unique FOV, which is that 51st column, converted it to a matrix, and then rotated it. That way our rows are our gene sets now, and our columns are our fields of view, which we can see down in the figure there is exactly how the matrix looks. Gene sets in the rows and fields of view in the columns. Next, since we specified that if we have less than n number of cells, 20 cells, we're going to return NA. In order to create a heat map where there's distances, we have to give it some kind of value. So I gave it a value of 1, which means that it's absolutely not significant. And then we can do a log transformation. So I did negative log 2, which means the higher the value, the more significant that it was. And I added this little fudge factor here because if we had 0 permutations that were less than our observed, we're going to end up with 0, and we can't take the log 2 of a 0. So by adding this and it being smaller than the values that we have, it's going to push up, and it's going to tell us very easily what is the significant values, and they stand out pretty readily here in our plot. And then negative, so greater is more significant, so on and so forth. So if we go ahead and plot that, we get this guy here, and we can see the rows are our gene sets and the columns are our fields of view. The fields of view are very smushed together, but here I think by looking at this global view, we can really see something with our fields of view. And we can see on the very left-hand side, maybe about left 50%, it's all very non-significant. We have some bands of no significance. That very blue is going to be probably those that had a value of 1, which would then be logged to 0, log 2 to 0. The red values are going to be those that were very significant, so they're standing out. But the left-hand side, and then we have the right-hand side being maybe 2 tissue niches. If we look even further, we can see maybe from 80% to the right, maybe even more than that, 85% to the right, there is 2 more potential niches depending on how we want to cluster this. We can see maybe one here, and then another one in these last handful of fields of view. So that is the gist of how to do the STenrich method of spatial gene set enrichment. Let me know if you have any questions, and thank you for watching.